The news for families facing a recurrence of Ewing sarcoma, this bone and soft tissue cancer in kids, it's just frankly devastating. The
Yeah,
odds of long-term survival just plummet because, well, our current treatments often just run out of steam.
they do. It's a huge challenge.
That's why the research we're digging into today feels so important. We're really hunting for new answers, aren't we?
Absolutely. When the standard approaches stop working, we urgently need fresh perspectives,
Yeah.
new vulnerabilities to target.
Uh-huh.
And that's precisely what this recent work from QRA Chi March 2025 is all about, really getting down to the fundamental mechanisms of this disease.
Okay, so let's get into it. We've got excerpts here from a scientific paper that takes a really close look at structural and functional analysis, I think they call it.
That's right.
And the star of the show seems to be this fusion protein, EWS-FLI1, and how it interacts with the very blueprint of the cancer, its DNA.
Exactly. Our goal today is to really understand the nuts and bolts of this interaction. We want to know exactly why this binding is so crucial for the cancer to thrive.
Right.
Because, that knowledge could point us directly to new ways to fight it, maybe with more effective, more targeted therapies down the line.
So this EWS-FLI1 protein, it sounds like public enemy #1 in Ewing sarcoma.
Well, yeah, in a vast majority of Ewing sarcoma cases, you find this very specific genetic glitch. The EWS gene and the FLI1 gene, they fuse together,
Okay.
and that creates this abnormal EWS-FLI1 protein. What's really striking and kind of unusual in cancer is that this fusion is often the only major genetic problem.
Really? The only one?
Often, yes, which makes EWS-FLI1 a really prime target. If we can figure out how it works, maybe we can figure out how to stop it.
And within this EWS-FLI1 protein, you mentioned there's a particular area of focus, the FLI1 part and its DNA binding domain, or WD. Why is this specific piece so critical?
Yeah, think of the FLI1 DBD as the business end of the EWS-FLI1 protein. It's absolutely essential for the cancer to develop and progress what scientists call oncogenesis.
Okay, at the business end, I like that.
Right. This domain is like a master key. It dictates exactly where the fusion protein attaches to the cell's genetic material. Without it, EWS-FLI1 simply can't exert its disease-driving influence.
Now, the research keeps mentioning these GGAA microsatellites. That sounds pretty technical. What are they in simple terms, and why are they so important for EWS-FLI1?
Okay, yeah, it does sound technical. Imagine sections of DNA where the sequence GGAA just repeats over and over again, like GGAA, 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 GGAA.
Got it. Just repeats.
Exactly. These are contiguous GGA DNA sequences, or microsatellites. And it turns out that EWS-FLI1 has a really strong preference for latching on to these repeated GGAA sequences.
Oh, okay.
Specifically in the promoter regions of certain genes. Think of promoter regions like the volume knob for a gene.
Oh, interesting analogy.
Yeah, so by binding to these GGAA repeats, EWS-FLI1 can abnormally turn up the volume on some genes or, mute others, which is what drives the cancer's growth program.
Can you give us a specific example, like a gene that gets its volume cranked up?
Yes, absolutely. The NR0B1 gene is a key example. Its activity gets cranked way up regulated by EWS-FLI1 binding to one of these microsatellites, specifically one with 25 GGAA repeats.
25 repeats. Wow.
Yeah, and this upregulation is absolutely essential for the cancer to thrive.
Okay, so EWS-FLI1 likes to grab onto these GGAA repeats, but the really fascinating part of this new research is that it's not just like acting alone, right? It's more of a team effort.
Precisely. That's the really important finding here. The FLI1 DBD doesn't just bind to these GGA sites one at a time. It does so cooperatively.
Cooperatively.
Yes, forming what the researchers describe as a nucleoprotein filament along the DNA.
Mm-hmm.
What's fascinating here is that when one FLI1 protein binds, it actually makes it easier for the next one to attach right next to it.
Huh. Like they're helping each other out.
Exactly. It's like they're helping each other get a better grip. So the key insight here is that EWS-FLI1 doesn't act alone. It's a team player.
Okay.
And this teamwork, this cooperative binding, well, that might be a potential weak spot we could exploit with new therapies.
Right. Interrupt the teamwork. So to really get a picture of this teamwork, the researchers used a technique called X-ray crystallography. What does that actually let you see?
Well, think of X-ray crystallography like taking a super high-resolution 3D photograph of incredibly tiny molecules. It's amazing stuff.
Wow.
So the researchers were able to create crystals of the FLI1 DBD actually bound to pieces of DNA containing 2, 3, and even 4 GGAA repeats in a row.
So they could see it right there attached.
Exactly. And these structures reveal the very specific ways that the FLI1 proteins interact with each other as they line up along the DNA. It's like seeing the actual molecular handshake between these
A
proteins.
molecular handshake. Okay.
Yeah, so the researchers essentially asked, how do we actually see these proteins holding hands? And their answer was this technique that allowed them to get this visual snapshot.
And what were some of the key structural details they saw in these molecular snapshots? What did the handshake look like?
Well, several really crucial details emerge. First, the parts of the FLI1 protein that directly touch the DNA, they call them recognition helices.
Okay.
From adjacent to FLI1 proteins, they form a specific angle relative to each other. So there's a geometry to it.
Right.
Second, they identified specific chemical building blocks, amino acid residues like tyrosine 341 and aspartate 333 on neighboring FLI1 proteins that directly interact.
So specific contact points?
Exactly. Think of these as the key points of contact in their handshake.
Were there any other interesting structural observations? Anything else that jumped out?
Yes, actually. The very end of one FLI1 DVD, what they call the C-terminal region, it makes direct contact with the next FLI1 DVD in line.
Ah, so the tail end connects.
Yeah, they even describe this region as having a kind of bracket conformation in its tail,
Yeah.
and it interacts with a specific loop called L23 on the next FLI1 protein.
Wow, that's detailed.
It is. And importantly, when they looked at the structures with 3 and 4 GGAA sites, they saw that FLI1 completely filled all the available binding spots, no gaps,
Okay.
which really reinforces this idea of a continuous filament being formed along the DNA.
So they got these amazing pictures of how they link up. But, I mean, how did the scientists prove this helping hand effect, this cooperative binding, wasn't just, how it looked in the picture? How did they measure it?
Right, good question. Seeing the structures is one thing, but proving the cooperativity, the functional effect, requires more. They use techniques like EMSA.
AMSA.
Yeah, it's like a molecular sorting experiment.
Yeah.
It shows how well proteins stick to DNA. And they also use something called fluorescence anisotropy. That measures how molecules tumble in a solution. And it changes when they bind to something bigger, like DNA. These experiments allowed them to actually measure how much the binding of one FLI1 protein enhances the binding of its neighbor, especially on DNA, with 2 adjacent kind of weak GGAA sites.
And what did those experiments tell them? Which parts of the FLI1 protein are most important for this cooperative binding?
Well, the EMSA data showed this really striking all-or-nothing binding when there were 2 weak GGAA sites.
All or nothing.
Yeah, meaning you either got basically no binding at all, or both sites were completely occupied by FLI1, almost nothing in between. That's a really strong sign of cooperativity.
I see. Because one helps the other so much.
Exactly. They also discovered that if they chopped off that C-terminal region of FLI1 we mentioned earlier,
The tail end.
right, that cooperative binding was significantly weakened, compromised.
Okay, so that tail interaction is definitely important functionally. What about other regions of the protein?
Well, interestingly, they looked at the other end, the N-terminal extension of FLI1. Initially, it seemed to act a bit like a brake, inhibiting individual binding.
Inhibit it, so making it harder to bind.
For a single molecule, yes. But surprisingly, it actually enhanced the cooperative binding when multiple molecules were involved.
Huh. That seems counterintuitive.
It does, right?
Yeah.
But the crystal structure of the unbound FLI1 DVD with this extension showed that it can actually fold back on itself and sort of get in the way of DNA binding.
Okay.
So the idea is maybe when other FLI1 molecules are present and trying to form that filament, this extension gets moved out of the way, or perhaps even contributes positively to those cooperative interactions between neighbors.
Fascinating. And they didn't just observe, they actually messed with the protein to understand these interactions better, didn't they, like mutations?
Exactly. They made very specific point mutations, changing single amino acids. They targeted ones they had seen interacting between the FLI1 proteins in their structural images, like N329E and D333G.
And what happened?
Well, the D333G mutation was particularly dramatic. It completely eliminated the cooperative binding, just wiped it out,
Wow.
which really highlights the critical role of those core protein interactions they visualize in the structure. It confirms the handshake is essential for the teamwork.
So bringing this all back to Ewing sarcoma, what are the really big implications of these findings for understanding and hopefully eventually treating the disease?
Oh, these findings are really significant. Because this cooperative assembly, this filament formation of EWS FLI1 on these GGAA microsatellites, it seems absolutely crucial for the abnormal gene regulation that fuels the cancer.
Right, the core problem.
Exactly. So by understanding the specific protein regions and the specific interactions involved in this assembly, the handshake, the tail interaction, we open up the very real possibility of developing drugs that could specifically disrupt this process.
Like a drug that stops the handshake.
Precisely. Or prevents the filament from forming in the first place. The researchers themselves point out that interfering with this assembly has already been suggested as a potential therapeutic strategy. This work gives much more detail on how you might do that.
So if they work together, maybe you target that glue holding them together or stop them linking up. That sounds like a very specific point of attack for a new drug.
That's the hope. Much more targeted approach than, say, traditional chemotherapy.
That's really promising. And is this just relevant to Ewing sarcoma, or could it apply elsewhere?
That's a good point. It's quite possible it's broader. The FLI1 DBD is very similar in its basic structure to the DBDs of other related proteins, other ETS family transcription factors, like a proteins called ERG, FAV, ETV1, ETV4. These are also involved in some Ewing sarcomas or related cancers, often as fusion proteins too.
Ah, okay.
So this suggests that the cooperative mechanisms they've uncovered for FLI1 might actually be relevant to a broader range of these cancers driven by similar fusion proteins.
This has been a really fascinating look at the FLI1 part, the DNA binding part. But what about the other half of the fusion protein, the EWS part? Does that play a role in this bigger picture?
Yeah, that's really the next frontier, and the researchers acknowledge that. The EWS region has what's called a low complexity domain. It's structurally quite different.
Low complexity,
It means it's often intrinsically disordered, less folded, and these domains are thought to be involved in forming higher-order structures within the cell, things like little droplets or gel-like structures, sometimes called condensates.
Condensates, like little functional blobs.
Kind of, yeah.
Okay.
These could further influence how the whole EWS FLI1 protein assembles on DNA and maybe how it recruits other important cellular machinery.
Like what kind of machinery?
Things like chromatin remodeling complexes, the BAF complex is one example, which help control access to our genes by opening or closing the DNA packaging.
Okay, so it gets even more complex.
It definitely does. But this detailed research on the FLI1 DBD and its cooperative binding provides a really critical foundation for understanding these more complex assemblies involving the EWS region as well. You need to understand the foundation first.
Right. So, to kind of recap the main points here, we've learned how the FLI1 DNA binding domain doesn't just act in isolation on these GGAA repeats.
No, definitely not.
It forms teams, cooperatively building these filaments along the DNA. And this teamwork is orchestrated by very specific structural interactions involving different parts of the protein, the handshake, the tail.
Precisely. And this detailed molecular understanding, while it delves into the very, very small, it gives us a crucial shortcut, really, to understanding what makes this challenging cancer tick.
Shortcut, yeah.
It's that aha moment, seeing how individual protein pieces come together to create this larger disease-driving machine. It's almost unbelievable sometimes to think that scientists can visualize something happening at that molecular level.
It really is, to actually see these proteins interacting.
Yeah. It provides such clarity.
It truly is remarkable how these tiny molecular details can have such profound implications for understanding and, hopefully one day, treating a disease like Ewing sarcoma. It really makes you think. Given these incredibly detailed structural insights into how FLI1 forms these filaments, what innovative therapeutic strategies could scientists now dream up to specifically target and disrupt this critical assembly in Ewing sarcoma?
That's the multi-million-dollar question, isn't it? But these structures certainly give drug designers a much clearer map to work from. It's definitely something exciting to ponder for the future.